Coordination, the linking system, part 6. Students, in our previous parts, we have understood how animals respond to different stimuli by coordinating their body parts. We have seen nervous system and endocrine system are the two systems which work hand in hand to bring coordination among different movements exhibited by animals in response to stimulus. Right? But what about plants? Does plants do respond to stimuli? Does plants exhibit any movements? If so, how their movements are coordinated? Does they have nervous system and endocrine system like animals? Let us see answers for all these questions in this video. So let's start our session with a small activity on Mimosa pudica. Students, have you seen this plant at any time? It is commonly called as touch me not. Most of you might have seen it. It's a small shrub that grows in gardens, croplands, open lands, and you might have seen even in your school ground also. This is a small shrub. If you have seen it, please go to that plant once again and do this activity. What you have to do is just touch the leaves of the plant. Are they folding? Your answer will be yes. If so means if they are folding, in which direction they fold? It is inward direction. Look here. This is before touching. After touching, the leaves, they get folded like this. How they got folded? It is inward. In the inward direction they have folded. Right? After folding, these leaves, they expose small thorns. And these thorns protect the plant from being eaten by predators. So folding tarvata chinna thorns anetivi bayatiki expose avutayi, and because of those thorns herbivores anetivi tinakunda untayi anamata. So this is the response that the plants are showing in response to touch. From this experiment, you can clearly say that plants do exhibit movements. Plants do respond to stimulus, right? But if you clearly observe here, though you have touched one leaf, the other leaves which are nearby, they also got folded. How is this happening? How this leaf is sending that information to other leaves? This is what is coordination. How this coordination is happening will be understood in this session. So, our next question regarding this activity is, try to give similar examples. So, similar examples means examples of movements exhibited by plants. So this is also another question under this activity. So 3 questions are asked. First one is, are they folding? Next is, in which direction? Next, you are asked to give similar examples of movement. So let's see what you can answer for this question. Movement towards light is called as phototropism. Stems exhibit this. Movement towards water is called as hydrotropism. Roots exhibit this. Movement in the direction of gravity or in the direction of earth is called as geotropism. Roots exhibit this. Movement in the direction of an object when touched, this is called as thigmotropism. Tendrils exhibit this. Tendrils are wiry structures that arise on stems of creepers. So weak stems unde చెట్లు ఉంటాయి కదా, పాకే చెట్లు, లైక్ కుకుంబర్ ప్లాంట్ కావచ్చు, బిటర్ గార్డ్ ప్లాంట్ కావచ్చు. సో ఈ క్రీపర్స్కి వైరీ స్ట్రక్చర్స్ ఉంటాయి టు hold the support, and those wiry structures are tendrils. Tendrils exhibit this response called thigmotropism. Response is nothing but movement, right? So they exhibit thigmotropism. Next, movement towards chemicals, movements in the direction of chemicals is called as chemotropism. Pollen grains exhibit this. Sufficient diagrams to illustrate these movements or tropisms are not given in our textbook, so I have brought some pictures from outside sources. Using those pictures, I explain these tropisms clearly later. Now the question is, how are plants able to do these movements? How are they able to respond to this stimulus? Do they have nervous system or endocrine system like animals? No, definitely no. They don't have nervous system and they don't have endocrine system. What they have then? They have hormones. Like animals, they have hormones, but we cannot call this hormonal system as endocrine system because plants don't have endocrine glands. They just produce hormones from cells. So we should not call this hormonal system as endocrine system. Here, the hormones are called with a special name called phytohormones. Phyto means plants, hormones means they are chemical messengers. So, what are phytohormones? These are the chemical substances which make the plant show responses to different stimuli like light, heat, water, touch, pressure, chemicals, gravity, etc. These are also called as growth hormones because they achieve this type of movements by controlling one or other aspects of plant growth. So we can call phytohormones as growth hormones also. Some of the hormones and their actions are given in this table. This table becomes meaningful after I explain some concepts. I borrowed those concepts from outside sources. Let us first go through those concepts and return back to this table. Okay, now our first concept is regarding opening and closing of stomata. You know that stomata are small openings present on leaves protected by kidney shaped cells called God cells. So see here, these are the kidney shaped cells and they are protecting the openings within them. And these openings are called stomata. These stomata are open during daytime and they are closed during night time. When they are open, carbon dioxide enters into leaves, then the photosynthesis occurs in leaves and oxygen is liberated. This liberated oxygen also comes out through these pores. Along with oxygen, water also comes out of the plant. This leaving water produces cooling effect and saves the plant from sunlight. This is called as transpiration. During night time, as sunlight is not available, no photosynthesis takes place, so stomata closes. This closing conserves water reserves in the plant. Now, hormone responsible for this closing and opening of stomata is abscisic acid. In other words, the opening and closing of stomata with respect to the availability of sunlight is coordinated by the hormone called abscisic acid. Now, our next topic is concept regarding leaf aging. Just as we age, leaves also age. These are the stages of leaf aging. In this group, this is the first. This is the young leaf which is rich in chlorophyll and is more capable of photosynthesis. Thus, photosynthesis occurs in large scale. More and more food reserves get accumulated in the plant due to this kind of leaf. The last member of this group is this leaf, and this is quite useless for photosynthesis. There is no chlorophyll at all. So, this leaf falls off. A hormone by name cytokinin delays this process of aging and prolongs the active life of the leaf. Under the influence of this hormone, leaves continue to be young and contribute more and more food reserves to the plant. Thus, plant produces high yield. Understood? So, the hormone that is responsible for delaying of aging is called as cytokinin. Hope you understood. Now, let us move on to our next topic, concepts regarding elongation, cell division and differentiation. Elongation is increase in the length of a cell. A hormone called auxin is responsible for this elongation. See here, a plant tip or plant shoot is bending towards sunlight. Right? This is because auxins have the tendency to move away from sunlight and go and accumulate on dark side. You know that auxins have the capacity of enlarging or elongating the cells. So, as auxins move away from sunlight and go to dark side, the cells there they enlarge. Understood? Compare the 4 cells on dark side and those on the light side. Those on light side remain normal, but those on dark side they enlarge. Due to this enlargement, the shoot bends towards light. Thus we can say enlargement of cells produces bending, and this bending is under the action of auxins. Next concept is cell division and differentiation. See here, one cell has become 2, 2 has become 4, and 4 has become 8. Like this, division continuously occurs repeatedly until it produces a required number of cells. After that, differentiation takes place. At this stage of maximum number of cells, the size of all the cells is identical in size, shape and size. This is because growth in them has occurred uniformly in all directions. But in differentiation, growth occurs at different rates along different directions. This produces cells of different shapes and sizes. These cells of different shapes and sizes become the parts of the plant, like buds, leaves, flowers, roots, etc. A hormone called cytokinin promotes cell division, but a hormone called auxin produces cell differentiation and also elongation. Did you understand? So, for cell division, the hormone is cytokinin. For cell differentiation and elongation, the hormone responsible is auxin. Understood? Now, our next concept is concept of dormancy, breaking of dormancy, which can also be called as sprouting. I explain this with reference to two diagrams given over here. See here, pollination produces seeds. Seeds undergo maturation for some period. They start germinating or germination when the environment is favorable. The period between maturation and germination is called as a dormancy period. Sometimes under the conditions of continuous rains, seeds tend to germinate, but we cannot plant them in the fields during rainy time. We have to use some hormone to prevent this tendency. Now, the concept of dormancy and breaking of dormancy is clear to you with reference to seeds. Let us apply the concept to the buds. See here, the lower two buds are dormant and the upper two buds are sprouting. If all buds grow at a time, it is a big loss to the farmer. So he uses gibberellins to break dormancy of the buds in some selected areas of his land and spray abscisic acid to prolong dormancy in certain areas. Nursery people use gibberellins to break the dormancy of buds and show some flowers in the plants they try to dispose to us. Understood? Now the last topic is sprouting of lateral buds when apical bud is cut. Here is a plant with an apical bud and many lateral buds. So these are the lateral buds. This is the apical bud. The terminal one is the apical bud. If we cut lateral buds, then the plant grows taller. But if we cut apical bud, the lateral buds, they start sprouting and many branches will develop laterally. This is under the influence of a hormone called cytokinin. Left to itself, that is, if we don't interfere with the growth of the plant, then the plant has the tendency to grow high under the influence of hormone auxin. This is called as apical dominance. But if we want shorter plants with many branches, then we have to prevent apical dominance by cutting apical bud. Understood? Usually we expect plants in our garden to cover more and more area and give us shade for a wider zone. So we have to cut, in that case we have to cut apical buds so that the lateral branches, they develop vigorously and produce shade for us. Now we are in a position to understand the table given in our textbook. Let us go back to it. This is the table. Here are the hormones and these are the functions. The first hormone is abscisic acid. For this, closing of stomata and seed dormancy are the functions. Next, it is auxins. Auxins are helpful in cell elongation and differentiation of shoots and roots. What does this mean? Differentiation of shoots and roots means some of the cells after differentiation they form shoot system and some they form root system. Next, it is cytokinins. Cytokinins promote cell division, sprouting of lateral buds. They delay aging in leaves and They help in opening of stomata. Next hormone is ethylene. This was not discussed before, but it is very familiar for us. In our market, we see mostly ethylene treated fruits. So if the farmer wants to dispatch a batch of fruits, then he use this gas, ethylene. Ethylene is a gas. He apply that gas to the fruits and ripen them instantly. So ethylene is useful in ripening of fruits. Next it is gibberellins. Gibberellins are gibberellins. Gibberellins, they help in germination of seeds, sprouting of buds, elongation of stems, stimulation of flowering, development of fruit, breaking of dormancy in seeds and buds. The context in which these are to be used has already been discussed. Remember that stimulation of fruits and flowers is necessary for cultivators, because if all the fruits or flowers bloom at a time, it is not economical for the farmers. So they have to choose different areas and use gibberellins in those areas to get flowers or fruits from that area. Understood? Hope this table is very meaningful now. Now let us move on to activity number 5, discovery of the hormone auxin. Hormone auxin is the first one that has been discovered. Let us see how it was discovered. Sow a seed near the wall of a jar. Then seeds germinate after 4 to 5 days. Then observe how the roots and shoots grow. Assume that this is the seed extracted from the jar. As shown in this picture, roots grow towards earth and shoots grow towards sunlight. Right? Okay. Now tilt the jar and keep the plant as shown in this figure. See here, like this. And answer these 3 questions given here. The first question is, does the shoot take a horizontal tilt after a week? No. In this picture, it has not taken. In fact, it has taken a vertical tilt towards light. So light is over here, so the plant is growing towards light. That is the answer. Next question is, which side of the shoot might have grown more and which side less to bring this effect? This effect means the bending effect, the tilting effect. It has tilted towards sunlight, right, or light. So to bring this tilting effect, which side of the stem has grown more, whether it is on the exposed side or the unexposed side. So when light is over here and falling on this side, the cells on the opposite side might have grown more, because I have explained this picture already, right? When light falls on this side, the auxins at this region have the tendency to move away from light and get accumulated on the dark side, that is unexposed side. So when auxins go there, what is the action of auxins? Auxins enlarge the cells or elongate the cells. So cells on the unexposed side, or we can call it as dark side, they enlarge. So as the cells at this region enlarge and cells at this region remain same, bending takes place. What is the answer? Unexposed part of the shoot might have grown more and exposed part less. Did you understand? Now, do you find any difference in the shape of epidermal cells? That is, the epidermal cells at the tip, they are asking. So look at this picture. Epidermal cells of exposed part are shorter than those of unexposed part. This is because auxins have the tendency to move from light side to dark side. That is what given here. Yes, difference is there. Epidermal cells of exposed part are shorter than those of unexposed part. This is because auxins, the growth promoting hormones, move away from the light exposed side, causing the unexposed side grow faster than exposed side. Understood? With this activity, you are now in a position to understand the experiments done by Darwin and Wendt in discovering auxins. Both the scientists have done their experiments on oat seedlings. Oat is a plant and seedlings are nothing but the sprouted seeds. So these are the sprouted seeds. When Darwin applied light to the seedlings of oats, they showed bending. See here, this is bending towards light. Then he cut the tips of the seedlings and he found no bending. So when tips are cut, there is no bending in the seedlings towards light. Later, he covered the tip with a metallic cylinder so that no light passes through it. Then he didn't observe any bending. Okay. Later, he has covered the tip with a transparent cap so that light can pass through it. Then he observed bending. Finally, he covered the base of the seedlings with the metallic caps. Again, he observed bending. From these observations, he has concluded that light makes some influence to pass from tip towards bottom, causing bending. So what is his conclusion? He concluded that light makes some influence, some chemical to pass from upper parts to lower parts, causing bending. This was his conclusion. He did not discover oxygen yet. Later, Wendt continued his experiments and he succeeded in separating or isolating the influence. He has isolated that influence. Let us see what is his experiment. These 4 pictures are given in our textbook. For examination purpose, draw them, but for understanding, look at these 5 diagrams. In the first diagram, the tip is being cut. This tip contains that influence, what Darwin has observed. It has some influence. Then in the second picture, the tip is kept over agar block. So this is the agar block. Agar is a polymer. It is a gel. It can absorb materials coming from the tip. Okay. So the tip is cut and is kept on agar block. In the third diagram, when the tip is kept, the oxygen present in the tip diffuses into agar block. We now know it is oxygen, but it was influence for Wendt. So this was influence for him, and we now know it is oxygen. So oxygens diffuse into agar block. This agar block is later cut into small bits, and one of the bit is kept over the stump. Stump means that from which tip is removed. So this is the oat seedling tip, oat seedling from which tip is removed. So that without tip is called stump. So now the agar bit which is cut is kept over the stump. After putting agar block, the stump is kept in dark room. After an hour of putting it in dark room, the stump started bending towards the agar, in the direction of agar block. See here, it has bended. From this, he has interpreted that the bending was in response to some chemical stimulus. Some chemical is being passed from the agar block into the stump, making it to bend. Later, this influence or this chemical was named as oxygen. So, oxygen was not the name given by Wendt, but it was later on named as oxygen. So, this is how the oxygen was discovered by the experiments of Darwin and Wendt. Hope you understood. So, movement towards light is one of the main response that plants make. Let us consider some other movements. These movements can be divided into 2 groups: tropic movements and nastic movements. In tropic movements, direction of movement of plant is determined by stimulus. Usually, movement is in the direction of stimulus itself, whereas in nastic movements, direction is not determined by stimulus. That is, direction of movement and stimulus are in different directions. Understood? We have already learnt 5 responses to 5 types of stimulus. Here, response means movement towards stimulus. So, phototropism is movement towards light. Stems exhibit that. Hydrotropism means movement towards water. Roots exhibit that. Geotropism, movement towards gravity or earth. Roots exhibit that. Thigmotropism, movement towards touch, and this will be exhibited by tendrils. Chemotropism is movement towards chemicals. pollen grains exhibit this. Now in next slide, we study this in detail. Look here. See here, seedlings are kept near a window. They started bending towards light. This is phototropism. Observe here, roots, however they are kept in part, they always grow towards gravity or earth. See here, they are growing down. This is geotropism. See here, the roots of these two plants are bending away from wall and moving towards water source kept in the middle. This is called as hydrotropism. If a plant is grown near a wall or around a rocky surface, then it tries to grow in the direction of water. That is, it seeks water through the roots. This is hydrotropism. Now the pollen grains. Pollen grains, after pollination, they reach a stigma. Stigma secretes sugary substances. It's a chemical substance. This chemical substance stimulates the pollen grains to germinate. After germination, they move towards ovule. So this response of pollen grains towards the chemicals released by stigma is called as chemotropism. Got it? Next, see here. It is a creeper plant whose stem is very weak. So it develops string-like structures, wire-like structures. These wire-like structures make the stem stand and grow erect. These spring-like structures, they grow and grow until they touch a surface or a support. Once they touch a support, then they wind around it and help the plant to move upwards. This is thigmotropism. This is seen in creeper plants like cucumber, bitter gourd, bottle gourd, etc., whose stem is very weak. So with this we have come to end of the video. Finally, at the end, let us conclude that phytohormones are responsible for control and coordination in plants, and these phytohormones can be classified into two major groups based on their action. Some hormones are promoting growth. They are growth promoters, and one hormone is inhibiting the growth, and that is growth inhibitor. So what are they? Auxins, gibberellins and cytokinins are growth promoters. Abscisic acid is growth inhibitor. So this is the end of the video and end of the chapter. Let's meet in next chapter soon. Thank you.